we're recording. Success.
We're recording. Okay, perfect. Okay, perfect. So this is chapter 2 of the BrainBee PowerPoint. I think Jordan is up first with presenting, and you could just let me know when you would like to.
Oh wait, can I?
Oh, okay. So you actually.
Wait, is it gonna work? Oh, it will work. Okay. Well, I can.
Let me know if you need me to control the mouse, but it looks like you got it. So
Fabulous. Hello, everyone. My name is Jordan Bartfield. I'm currently a GREP student at the Mayo Clinic in the Department of Neuroscience, and also Vice President of Brainwaves, helping Gabby out with BrainBee. And today I will be presenting on sensation and perception for, okay, they will be deleted after 90 days. So we'll need to figure out some workaround for that.
okay.
But
Yeah,
I'll be presenting on sensation and perception as part of our BrainBee prep material. So when we talk about the term perception, okay, there's weird lags, sorry. This is not working. I apologize. Okay, I gave up control. Can you click through for it?
Yes.
Okay, sorry about that.
Maybe it's because I was moving the mouse while you were doing it and I didn't realize it would affect you. I'm sorry.
I think those of us who are in control of the PowerPoint, that wasn't working.
Okay.
So when we think about perception, what we're really thinking about is the way that our bodies interpret the experiences that we're having. So how do our bodies take in light and create vision? How do they take in sound waves and create noise? How do we take in temperature and create our sensation of temperature, or physical objects and create feeling? So that's kind of the idea that we're discussing when we're talking about perception. And so can you do next slide? We will be discussing specifically today vision, hearing, taste, and smell, and touch. But when people talk about your senses, there's actually a huge amount of debate over the number of senses. So some people will say there's 5 senses, and then other people like to get really technical, and they'll say that number is as large as 17 senses. And so today we'll be just focusing on these, but there is, areas of contention. Some people like to get very granular and say that your sense of heat is a specific sense and your sense of cold is a specific sense. So it's actually quite an interesting field of research and something interesting to look into. But our 5 today will be vision, hearing, taste, and smell, and touch. And so we're going to go through a few key terms to do with vision to begin with, starting with transduction. Next slide. So transduction is how we take in the energy and transform it into a perceptual stimuli. So I often think about signal transduction in terms of vision, because that's how I originally learned it. And in that way, the way that works is that we're receiving light waves to our eyes, and we'll go through the particulars today, but light waves come into our eyes and we're able to transduce those and turn them into a tangible stimulus, which, or I guess not tangible, but to us, that's the world that we're perceiving. And so what we have an energy stimulus, and that's turned into an electrical potential, and then it's carried through this network of cells and fibers to different areas of our brain, and then we process it and integrate it, and we have our vision. Next slide. So humans have binocular vision, and we have a lovely diagram of vision here. And one thing I like to point out whenever this diagram comes up is the idea of blindness. So in talking about blindness, a lot of people assume that when I say someone's blind, it means their vision is completely black. And if you were to survey 100 blind people, you'd actually get maybe not 100, but a large number of different experiences that they're currently having with regards to vision. So blindness can mean that someone has nothing there and they're seeing completely black. It can also mean that things are super blurry. It can also mean that they have difficulty with perception and have limited abilities in certain, sorts of light. And you can also have blindness at multiple different areas in the visual path. So here we have our eyeballs, and then these are our optic nerves going down, and our primary visual cortex is back here. And so if you have damage at any of these different points of the visual tract, you can have blindness, but blindness can result from a number of different causes. So binocular vision stems from our two eyeballs, and what happens is we have images coming in from both sides, and it creates a cohesive field that we perceive. So we don't have this kind of jarring disconnect between one eye and the other. As you can see, there's a nice gradient of vision where you're not seeing the switchover, but it is there, and it becomes very apparent when you have monocular vision. So we want to do, I think the next slide is monocular vision. Oh, no, it's not. So monocular vision is vision from one eye. And so if we were to cut out this eyeball, then everything here would be, can they see my mouse and yours?
I cannot see your mouse.
Oh, I've been doing lots of gesturing. That's so sad. If we were to cut out one eyeball, then our visual field would become very limited, and it would become much easier to appreciate where the distinction between the two fields are. So, okay, now we'll go to the next slide. So, hopefully you guys have gone through the neuroanatomy presentation that depending on which school you are, you had a formal, lecture on it. And if there are any questions about neuroanatomy that come up during this lecture that aren't clear, please feel free to reach out to us with questions so we can clarify information. We want to make this an easy and accessible experience for you all. So the thalamus is our sensory relay center. So I like to think of it as a train station where all of our senses are coming in, and then our brain takes that information and it sends it back out. So it's sitting right here. It's highlighted in a nice mint green. And oh, I keep forgetting they can't see my mouse. So when sensory information comes in, and that's going to be light in the form of electrical potentials, or sound will be coming in from your ears as sound waves. It'll have been transformed in some way before it reaches the thalamus. So you're not receiving a sound wave, actually, you're receiving a signal in the form of electricity inside the brain. But all of that information comes to the thalamus, and then the thalamus will send it back out to different parts of the brain that help to incorporate that information. So that's the third step in transduction that we saw in the earlier slide. Do you want to go to the next slide? So when we have vision, the way it works is we start with the cornea. So the cornea is highlighted here. I guess it's not highlighted, it's pointed out here. So it's the most external part of your eye. And that's where light comes in first. So light is going to enter the cornea, and the cornea has multiple purposes. So as the exterior portion of your eye, it also serves as a protective barrier, just like skin. It's keeping things from coming inside. But light is going to enter, and then it will travel after it does some minor processing. So the cornea has some limited ability to kind of funnel light to a degree and sharpen it, if you will. Next slide. Then it will hit the pupil. And so we have our pupil here, this dark spot in the very middle, and then your iris, which is the colored portion of your eyeball. So when we're looking at someone's eye and we're saying you have green eyes, you have blue eyes, we're talking about the iris. And both the pupil and the iris work together to funnel light, and they can contract or expand in different ways. So if you've ever been to the doctor and you've had them shine a bright light in your eye, what they're looking for is they're looking for a reaction from your pupil. So they want to see your pupil changing in size. And movement of your pupil is controlled by some structures in your brainstem. And when a patient comes into the hospital and their eyeballs are no longer reactive to light, that's a sign to the physician who's treating them that there might be damage in the brainstem, because they're no longer able to have that functionality in their eye. And so your brainstem also has structures that control your breathing and your heart rate and all of the really necessary vital functions of the human body. So anytime a patient is coming in and they're no longer having light reactivity in the eye, it's what we call a fixed pupil. So you shine a light in their eye and there's no change in the pupil. That's kind of a big code red sign of, okay, something's going on. We need to mobilize everyone and figure it out. It's a good flag for danger. And so the pupil and works to kind of contract that light further. Almost, you could think of it as a funnel. So light starts as your cornea and it's way out here. And then it's in the pupil, and the pupil's helping to kind of contract it and concentrate it into a smaller area and direct it to the right spot in the back of your eye. Next slide. Here we have the iris, which is that colored portion that we were talking about. And so this term pupillary light reflex, that is the, did something happen? Okay. That is the light reflex I was just talking about that physicians will look for in patient's eye, and that it's a very big sign if they're not seeing one. And so the purpose of the reflex is to respond to light, and it allows us to, if we have a large amount of light coming in, so if someone shines a flashlight in your eyeball, you don't need that much light to see. And so we don't want that much light. And so it'll help to kind of keep some of that light out. Whereas if you walk into a room and there's absolutely no light, then we need all of the light we can get to help us see. And so it's going to expand and allow more light in to let us see better in more limited conditions. So here we have the official term. So dilating, where we're making the pupil larger to allow light to enter, and then constricting to make the pupil smaller to lessen the amount of light entering. And anytime we define a term, there's a good likelihood that it's important that you know the exact definition. So, you know, sometimes we'll talk about concepts and we'll discuss them, and they won't necessarily be, you know, written out on the PowerPoint. And those are terms that, you know, it's interesting and it helps to kind of give us context to the ideas that we're talking about. But, you know, it might not be tested material. But if it's on a PowerPoint slide, then it's highly encouraged that you know those terms. Okay, next slide. Okay, so here we have the lens. And again, all of these different structures are working together. So we started at the cornea, and then we have the pupil and the iris that we just talked about. And they're all working together to achieve this goal of bending these light rays to reach the point that we want them to go to. So it's almost kind of like when you were a kid and you had a hose and you stuck your thumb over it to shoot the water out in different ways. We're kind of doing a similar thing with light, where we're contorting these light rays, just like you did with the hose in your thumb, to shoot them to the back of your eyeball, which is where your retina is. So that's this structure at the very back. We can kind of see it shaded. We'll be talking about it next. But the lens is going to be focusing the light on the surface that it's located on the inner surface of the eye, but the light is going to be bending to further focus that light that we've been talking about. Next slide. Okay, here is the point that we've been wanting to reach this whole time with light is the retina. So all of these structures leading up to this are just trying to get the light into the correct spot on the retina and the correct amount of light. Because the retina is a kind of a picky bodily area, and so it's accepting of kind of a limited range of stimuli. So if you have way too much light, then the retina is really not going to be able to form much of a picture. If anyone's ever shined a flashlight in your eye, you know, you can't really see much. And if you have too little light, then you also aren't able to see anything. And so it's because the retina has this range that it responds to. And so all of these structures of the eye work together to kind of try to funnel that light into this area and achieve that range that the retina will respond to. And so here we have a picture of the human retina. If you've ever been to the ophthalmologist and they've made you stick your eyeball in your head into this big machine, and you have to stick your chin on the chin rest, and you open your eye up really wide, and they're like, we're going to shine a really bright light into your eyeball, and then it's going to go green. Please don't blink. This is what they're looking for. And so here is the picture that they get to look at when you do that. And what else is on this slide? So can we go back to the very first visual map? I think it's good to have a visual to discuss this idea. Yes, this one. Okay, so here we have light coming in, and can you see this if I do that?
Yes, I can.
Okay, fabulous. So if we have light coming in, so here's our little light ray, and he comes in and he hits this part of the eyeball. So that's going to go here, and it's also going to go here. And then they travel down. So here's our eyeball. Here we have our corneas, and then they come through the corneas. They get And this is a little bit confusing because it looks like it's coming around. What's really happening is the light's going to be funneled in, and then you have your retina right here, and it's going to hit. And then the important part I want to talk about is this divergence. So we have images coming in, and this is our left visual field, and this is the right side of our brain. So images come into your left visual field, and they're going to get mapped onto your right visual cortex. And images come into your right visual field, and they get mapped on the left visual cortex. So there's this crossover point. And this is called your optic chiasm. And when you dissect a brain, it's actually really cool because you can go in and you can actually look and see the crossover point and see where it jets back out. And the underside of a brain, depending on the section you're on and what's been done to the brain, you can get a very nice clean cut where you can see the crossing and this funneling, which is fun to look at. But and we will have a neuroanatomy section at the Brain Bee competition where we will be looking at, I believe sheep's brain is what we are going to be using, but we'll be taking those, and you guys will be given a worksheet with little lists of different parts, and there'll be brains all around the room, and you're going to get to pick out and say, okay, this is this part and this is this part. So you guys will get experience with brains. Okay, so can we go back to where we were?
Yes, and I'll just add that we'll have a lesson plan that goes over a lot of the neuroanatomy. And so don't worry if it's like overwhelming right now to think about, but yeah, we'll have a lesson for that.
One of the nice things about having this recorded is that students will be able to go back. Oh, I didn't realize that the annotations would not disappear. Oh, they're not clearing. Okay, here we go. I think we're good. Okay, one more. One more slide. There we go. Okay, so again, talking about our retina, and here's that optic nerve. So you can see these, that's where light comes out the back of your eye. And what's really fun is that there's a test you can do where you take a piece of paper and it has a dot on it. You bring it very slowly up to your face. And at some point that dot's going to disappear. And the reason for that is because there's a hole in the back of your eyeball and in your visual field. And the reason for that is because there needs to be a way for those nerves to exit the eye. So, all of this light comes into your eyeball and we funnel it all the way to the retina, and the retina receives it. And we're going to talk a little bit in a minute about how signal transduction occurs and how that light becomes a signal, but that signal needs to leave somewhere. And so it leaves through the optic nerve out the back of your eyeball, and the space that is dedicated to the exit of those nerves creates a hole in your visual field. But it's one of the many perceptual blindness features that humans have where we just don't notice it. So there's your brain is very good at filling things in. And so one of the fun things that I learned in college and came to really appreciate while getting my degree in neuroscience was the understanding that our perception is not reality at all. So your brain likes to have a complete picture of the world around us. And so it'll do a lot of tricks and different things to kind of fill in the image, and it'll trick your brain when there's a problem. And so that's why a lot of times people won't realize that they're having issues that have to do with the brain, because the brain fills in the world around us and does not allow us to recognize that those issues are occurring. And so can we go to the next slide? Oh, I thought we were going to talk about that. Okay. So here we have the optic nerve that I was just talking about. And so here we can see it coming out of the back of our eye. And oh, here is the diagram I was talking about. And so we're able to kind of see our optic tracks and then the optic nerves coming from the eyeballs. And so here is the underside of the brain. And then this is damage causes vision loss. But again, I think it's really important to recognize that damage at any point along the optic pathway can cause vision loss. So if you have physical damage to your eyeball, that can cause vision loss. If you have damage to your nerves, that can cause vision loss. If you have retinal damage, that can cause vision loss. People can have all sorts of different causes of vision loss, and it can have the same end function of you're not able to see, but the beginning cause is very different. Next slide. Okay, so now we're going to talk about types of neurons. And this was can be a little bit tricky to appreciate. But so here we have, I'm gonna do my little pointer. Can you see this mouse now?
I don't see a mouse.
Okay, then I'll just do. You see that now? You see this?
Yes, I guess.
Okay, so here we have our eyeball down here. And this is the retina that we've been talking about. He sits at the back of our eye. And again, here's our optic nerves that are leaving the eye. And so this little, see, now I've made it muddy, no one can see it. This black box is actually, so this large chunk is this black box expanded. So when we think about the retina, and can we go back 2 slides wherever we last looked at the retina up close? More. Okay, this one. So when we're pointing at the retina, we're pointing at this very thin slice. And it's really kind of, it covers the whole back of your eye, but you see this, and then can we go forward too? And so when you see this, it's kind of hard to appreciate the fact that you have all of these little structures shoved in there, but they're there. And your retina has several layers. Each of them have a different function, and they all work together to take the light rays that we had coming in. So we have a light ray, and it comes in, and it goes and it gets filtered. And so we have all of these light rays, and they come in, and they're filtered by our cornea, and then our lens and our pupil, and they all contract, and they get sent to the retina, which is back here, and it consists of all of these guys down here. And what the retina does is it takes in those light waves and it transforms them into an electrical potential that our brain is able to understand. So we're transducing it into a form that our brain can appreciate. So in the retina, we have 3 different types of cells. We have our neurons. We have our photoreceptors, our interneurons, and our ganglionic cells. And they're organized into these different layers. And they all communicate with each other. And there's lots of good resources where you can work through the different layers of the eyeball. And we will find some and attach them with when we send out this PowerPoint. But I think it's important that you guys gain an understanding of how all of these different structures work together, which we don't have time to discuss today, but I highly recommend some reading. And I think BrainFacts has some good resources on discussing the individualized different roles of these cells and how they all work together to create this phototransduction pathway. Next slide. So our ganglionic cells and our interneurons their main role is in processing and relaying information from the photoreceptors, but they don't respond directly to light. And so here we have, these are all the same diagrams, just put together in different formats. Here we have our optic nerves. So this is going to be coming eventually out the back of our eye. And then we have our ganglionic cells, which are closest to the back of the eye. Our bipolar neurons, our rods, and our cone cells. Another really strange thing about vision is that when you learn about vision, you learn optic nerve, ganglionic cells, bipolar, rod, cone. That's a very common fashion or order to learn it in. But what's actually happening is the opposite. So it's going to be perceived by our rods and our cones and our bipolar and our ganglion, I believe, and then go out the optic nerve. Next slide. Here we have our photoreceptors. So these are our rods and cones. If you guys have taken a psychology class, then you've heard of these. These are in charge of our color vision and also our black and white vision. We have the fovea, which, can we go back to the retina slide? Because we had a really pretty picture of that. So here, oh, no, okay, that works too. So here's the fovea. And it is where all of our cones are concentrated. I believe that's right. Yes. So, and then, so we have a large concentration kind of at the center of our eye. It's hard to orient ourselves on this image, but this is a picture of the back of your eyeball. This is your retina. And so light is coming in here and hitting, and then these cells are all leaving through this nerve. So this is kind of, this aspect is likely below this one, if that makes sense. So your pupil is probably somewhere around here, and then this is way behind. I hope I visualize that well. Okay, back to the slide we were on, the rods and cones. Okay, so here we have our rods. And the purpose of rods is that they allow us to kind of see in black and white, is the best way to think about it. And they're centered on the outside of our eyeballs primarily. So your fovea that we just looked at is where all of your cones are, and they're right in the center of your vision. And then your rods are all the way on, kind of circling around. And you have some dispersed inside of the fovea and closer to the fovea, but for the main part, they're going to be spread out all across your eyeball. And they allow us to see in very dim light. And one fun exercise that I like to do with my students is that I have them take 2 pencils and they hold them, and you guys can all do this at home. You hold them behind your head and then you slowly move them forward, and till you can just barely see it in your periphery. And then I ask them, okay, what color is it? And what I'm trying to get at is that what's happening is you have your rods and are in charge of your peripheral vision, because they're out here, but they're only for black and white vision. And so they don't encode color. And so if you're seeing something in your periphery, you actually cannot see the color of it. But like we were talking about earlier, we have this perceptual blindness where our brain just fills in the picture very effectively. And so we're not aware of these deficits we have until we do fun little tricks like this one, or the one I was talking about with the piece of paper, that force us to recognize those deficits. Next slide. Okay, so here's our cones. Like I said, these ones are responsible for our color vision, and they are extremely sensitive to light, and they allow you to see in very dim light. And so, you know, obviously when we look at the world, we're not seeing just 3 colors, we're seeing a whole variety of colors, and that is because our 3 colored cones will fire at different intensities, and different numbers of them will fire to create the colors we're seeing. So just like if you go into art class and you're mixing different colors, you know, if you want yellow, sorry, not yellow. If you want to create purple, you'll have to mix red and blue. And so our brains are doing that constantly. But, you know, it's quite fun to kind of think about all of these different things that are happening instantaneously, because, you know, when you turn your head into a room, the picture doesn't load like it might on a computer. It's pretty instantaneous, but a lot has to happen for that to occur. The light has to come in, your eyeball has to filter it, your retina has to receive it. We have to transduce the signal and turn it into something logical that has to be sent through our optic tract. It has to make it to the thalamus. The thalamus has to send it to the occipital cortex. And, you know, all of these things happen. And then we have vision. And so I think that's something really amazing that we forget to appreciate, is how many things have to go right for us to see and hear and speak and all of that. Next slide. Okay, so here's the macula and the fovea. So the fovea, like I said earlier, it contains only our cones, and it's super, super densely packed. And it's located right at the center. And so one fun exercise, if you go like this, you're kind of blocking out your fovea, because, you know, on the diagram we saw earlier, your light gets funneled in. And so all of kind of this area right here is coming into your fovea, and then everything around it. And there's a lot of overlap between the rods and the cones. They work together to create an image. But if you go like this, you're going to be blocking out, and you'll only be accessing your peripheral vision, which is rods only. And then we have our macula, which is where we have the best visual acuity. And that's partly because the light is coming directly to it. And so it's hitting it. It's being received at that point most intensely and most accurately. So it's not receiving kind of peripheral light, if that makes sense. And it's right around our fovea. And so here we have our fovea, and then right around it our macula. And if anyone's ever heard of the term macular degeneration, so if you have a grandparent who has some trouble seeing and someone's thrown out, oh, they have macular degeneration, that's, you know, what that is you have this portion of your eyeball is degrading. Next slide. Okay, this is just another neuroanatomy, or not neuroanatomy, eyeball anatomy. Next slide. And they have, Gabby, I want to just confirm that they have access to all of these PowerPoints. We'll be sending the PowerPoint as well.
Yes, yeah, they'll have access to all of this.
So I'm gonna go till 6, and then at 6, why don't we switch over to Sebastian's chapter? Just so we can try to cover both chapters, even if we don't get through all this.
Sure, sounds good.
Okay.
Well,
So
I guess up to Sebastian too.
Here's our optic chiasm, and this is that crossover point that I was discussing earlier, where you have a junction where both the fibers from both eyeballs cross on their way to the brain. And so this is where the information of from your left eye is going to be sent to your right eye, and information from your right eye is going to be sent to your left eye. That's where that happens. Next slide. These are some great figures we have. Okay, I think that students will need to go through and because of time, why don't we jump through the specific cells? Keep going. Okay, here we have the geniculate nucleus. It plays a role in visual processing in the thalamus. So earlier I discussed how the thalamus is like a train station where, you know, sensory information comes in and then gets sent out. And you can think of this nucleus as kind of one of the tracks on the train. So if you walk into a train station, you know, imagine Grand Central Station in New York, and there's trains going to, you know, Michigan and Maryland and Virginia and all over the place. And so the geniculate nucleus is our train that's going to be sent to our visual cortex and help to relay that visual information to where it needs to go. And our medial geniculate nucleus is going to send information to the auditory centers of the brain. And one really good way to think of it is lateral geniculate nucleus starts with an L, and L is for light. And medial geniculate nucleus starts with an M, and M is for music, is how I always remembered it. So medial geniculate nucleus is going to handle hearing, and lateral geniculate nucleus is going to handle light. Next slide, please. Okay, so here's our visual cortex, our primary visual cortex, and that's going to be located in our occipital lobe. And it's where information is from both eyes is going to converge onto single cells. So up until this point, the information from each eyeball has been traveling kind of parallel to each other and then crossing over at the chiasm, but not meeting. And here it's going to meet and create the picture that we perceive. Okay, next slide. So why don't we skip forward to, I'd love to try to hit hearing. We want to skip to, what time is it? 5:44. Okay. Oh, this is fun. Visual disorders. And so earlier we were talking about, so obviously this is not a form of blindness, but earlier we were talking about how you can have many different ways to cause blindness or just vision impairment. So, you know, a lot of times when you see someone out and about who is using a cane or a guide dog or some other sort of visual aid, they might not actually have complete vision loss. So in order to necessitate those tools, you don't need to have complete vision loss. You just need to have significant vision loss. And so that can be generated in a number of different ways. And so cross-eyed, I don't know if anyone ever told you that if you crossed your eyes, they get stuck that way, but I would like to be, hopefully not the first, but I would like to announce that that is not true. There is no rubber band, it will not snap. You can cross your eyes all day long and your eyeballs will not stay that way. However, some people do have this problem, and it's called strabismus. And what happens oftentimes is that they begin to favor one eye over the other and they lose vision in one eye. And so one treatment that we have come up with to stop this is using eye patches. So it's surprising for some individuals, but we patch the good eye. So whichever eyeball the brain is favoring, we cover that one up and say you can't use it. And we don't do it constantly. So little guys will as early as, I think, 3 months old, maybe. It might be a little bit earlier, but they'll start patching and they'll do maybe 30 minutes a day and then maybe an hour a day. Or they'll do 30 minutes, 3 times a day. And so they work their way up, and you'll have kids who are in like 3rd or 4th or 5th or 6th grade who'll be patching for maybe 5 hours a day. And so what that does is it forces the brain to use the pathways that it was kind of neglecting before, and it maintains vision in that eye. I don't know if you've ever heard the term use it or lose it, but that is a very relevant term in terms of our brain's capacity to do things. So if you have a function or an area of the brain or pathways that are not at all being used, they'll rapidly start to kind of deteriorate, and other areas will encroach on them. And so if we don't maintain activity in those areas, it's kind of like a good roadway. If someone's not driving on it every day, things start to happen, and all of a sudden it becomes harder to drive. And then people say, oh, I'm going to take a different route. And so with the brain, we want to force our brain to keep using these different pathways. And so this is one way we treat that condition. Next slide, please. Here's macular degeneration that we were discussing earlier. So we do know some risk factors are smoking, obesity, and diet. So eating a healthy diet, not smoking, staying active are all good things to prevent this, but we don't have a good understanding of why it's happening. And here's a lovely depiction of, a pretty good depiction of what happens with macular degeneration. So we can see, there's these spots forming in the macula. Next slide. Okay, now we'll be talking about hearing, which is another fun topic. Next slide. So there's, when we talk about noise, there's a lot of different things we can talk about. Today, we're going to be talking about pitch, loudness, duration, and location. So when you think of hearing, these are all really important qualities of sound to consider. Next slide. So the way our processing of sound pathway, and all of these sensory pathways we're talking about are going to be important for you guys to know and understand and be comfortable speaking through and talking about and identifying different, anatomy related to different aspects of the pathways, and also what each happens at each step of the pathway and how they function. So sound waves are going to come in, they're going to vibrate on our eardrum, our tympanic membrane, and they'll travel through the cochlea to their auditory receptor cells, and those guys are going to bend. And this is the point at any hearing talk where I like to say that there's a limit to how far and how much and how often they can bend. And so you have little hair cells in your ears, they're called cilia, and they bend in response to sound waves. And you can damage them very easily, and they do not recover. And so that is why your mom or your dad or your grandma or your grandpa or your cousins and friends say, don't listen to music so loud. It's because you're aggressively bending those hair cells, and there will be a point at which you bend them so much that they're not going to be able to respond as effectively. And when they can't respond as effectively, they cannot help in that signal transduction pathway. And what happens then is that you have hearing loss. And so that's how really loud noise for a really long time causes hearing loss, is it bends the hair cells in ways that they're not supposed to, or super aggressively. So that's what's happening there. So that's why it's important to make sure that you are listening to things at reasonable levels. If your friend can hear it, then you should lower your volume, is the metric I like to go with. Next slide, please. So some key ideas in terms of hearing. Sound is derived from vibrations. So sound waves travel through the air. They can also travel through water or metals. And they can be described by frequency, temporal variation, and level. Then we have frequency and pitch. So these are topics you might have discussed in physics class. Frequency is the number of wavelengths that pass a fixed point in a unit of time. So we draw a little picture and we have our sound waves. Where's my little guy? So if 6 waves pass in 6 seconds, then it's 1 wave per second. And then that's measured in hertz. And then pitch is our perception of the frequency of sound waves. So I hope you all have seen a diagram of light rays. And so when we see a diagram of light rays, we see, you know, we have red light on one side and purple light on the other. And just the same way we have light rays of different lengths and frequencies, we have the exact same thing with sound waves. And so sound waves, if they're super fast and close together, they're going to be much higher. And if they're long and slower, they're going to be much lower. And so that's the same way we have super fast and high as one color, super slow and long as another color. It's the same idea with pitch. Then intensity and loudness. Intensity is the pressure exerted by sound waves on the tympanic membrane. And so, you know, kind of like how hard are those sound waves hitting that membrane? And that's how we perceive the intensity of noise as loud. Next slide, please. Okay, so these are different anatomy parts of the ear, which we will hit as we work through. Next slide. Okay, so we also have, Gabby, in this PowerPoint, taste, touch in here, and smell, right? Debbie?
My unmute button was not working.
Yeah.
So we have hearing, yeah, and we have the different, we have pressure waves, making sense of sound, a little bit about taste, touch and pain, sending and receiving messages. Yeah, I think, sorry, I forgot what slide we were on.
So I think, so the students will watch this PowerPoint. I think, if anyone has any questions, unfortunately, we just don't have the time to get through all the material today. I'm hopeful that when we meet with students next month, they'll have reviewed the PowerPoint and come with questions. So we can hit certain areas that people are struggling with.
Yeah, and I agree. I think maybe because it'll be kind of impossible to get through everything in 2 hours.
Yeah.
You know, all the different slides that we have. And so we could use it as a review for the students if they have any questions. And if they don't have questions, we could just go through the slides as we did. So I think it definitely will be better to serve as a review. So I agree.
where can we access?
And all this information is in the Brain Facts book, and you know, maybe we just add a few clinical topics here and there as well. But all of the rest of this is in the Brain Facts book.
Okay, are you talking about these PowerPoints? The PowerPoints are all in our folder, and we're also going to be providing them to the students ahead of each lecture.
Yeah, so it's so Aina, are you a student from Stanton or Mandarin, or are you from a different school?
You're from Stanton. Oh, I didn't know we had any Stanton prep students with us today. Welcome.
So I believe Miss Krieger uploaded the PowerPoints to an online platform. If she hasn't, I can go ahead and I may have forgotten to share the PowerPoints, but I believe that she uploaded them to the some online platform that you all use.
Does Ina, I didn't realize, I'm so excited that we have a student. Does Ina, I hope I'm saying your name right, or is it Ina or Ina?
Yeah, it's Ina, you got it right.
Okay, good to know. Do you have any questions on sensation, perception?
No, nothing so far. I mean, the visuals definitely helped out, so no, nothing so far.
Okay, so we will be giving you these PowerPoints. It sounds like you do not already have access to them. But your teachers have our emails, and they can provide those to you. Did you make these visuals?
A lot of them I've kind of put together, and some of them on Byrender have some templates, and so I took from those and modified some, and then kind of adjusted them in the PowerPoints to kind of make the point come across better.
Perfect.
But you are welcome to reach out to us with any questions, and we're happy to clarify any ideas. I know this is a lot of information. I actually did Brain Bee when I was in high school, and it was a lot of fun, but it was intense. But we're very excited to be having you participate.
Thank you guys so much.
And thank
Absolutely.
you for joining. We'll make sure that we get those PowerPoints to you guys, and I'll send the PowerPoints for next month ahead of time as well.
Okay, do we want to transition over to our movement guy?
Yeah, I'm happy to go over movement and at least a little bit. So,
Perfect.
totally down.
Would you like me to share the PowerPoint, or would you like to share it on your end?
I can share it on my end. Hopefully that
Okay,
results in a
perfect.
little bit less lag, a little bit more control.
Yeah, I think I just made you co-host, so you should be able to share.
Okay, thank you very much. Do you guys hear me well, by the way?
Yes.
I just kind of put on
Yes.
this microphone, but I was having troubles with it earlier. Okay, great. Is my screen showing? Are you guys seeing the bottom screen? Let's see.
It's
Maybe
not
I
in
should-
presenter mode.
Yeah,
I
I
see
see
the
everything.
notes section. You may have to switch it in display settings.
Yeah, I think it's this. Let me just duplicate it. There you go. Okay, great.
Oh, it's still
So,
showing for me in, oh, there we go. Okay. Maybe
yeah, I
there
think
was
it's
a
just a
lag.
little bit of lag. Yeah.
Perfect, thank you.
Yeah, awesome. All right, so chapter 3 is about movement. My name's Sebastian. A little bit of background why I wanted to teach this is because I was a personal trainer for some time, and I taught human anatomy at UCF, and so movement is something that I really enjoy teaching about, and obviously neuroscience and that stuff. So we're going to talk about reflexes. We're going to talk about voluntary and involuntary movements and how that pertains to your everyday, as well as certain pathologies that are important to understand if you want to, for example, have a future in medicine, if you want to have a future in the research behind these diseases and conditions. And so let's just kind of get into it. So movement is, we're able to move really because of our central nervous system coordinating through our peripheral nervous system with the rest of our body, specifically with our muscles. So whenever we talk about movement, we have to think about a couple of things. One of those things is your nervous system, both your central and peripheral nervous system, as well as the bones that make up your structure, your physical structure that act as levers. So right now I'm moving my hands. Hopefully you can see my screen. But we have bones all over the place, which kind of act as our structure, and then the muscles that pull on those bones in order to engage in an action or in a movement. So your central nervous system, your brain, your experience, you are coordinating all these movements, sometimes purposefully, sometimes kind of innately without thinking about it. And there's also times where we move where we don't have to think about it, and those are called reflexes, and we'll talk about that. So just a reminder, your central nervous system is made up of your brain and spinal cord. And then your brain and spinal cord will work together to send messages out through these connections to the rest of your body called peripheral nerves. So that's kind of a little bit of a basic overview. So when we were talking about voluntary movements, you might come up with a couple of things where, for example, I loved basketball when I was in high school. So when I was in your shoes, I was eager to go shoot a basketball, eager to run across the court and catch a pass and then do all those things. Those are voluntary movements that I choose to do, that I chose to work on. And that happens through conscious act, right? You want to reach for that glass of water. You want to put the basketball in the hoop. And so that is done through thoughts and then actions through that nervous system to muscle connection. By the way, that connection between your peripheral nervous system and your muscles, there's a junction there called the neuromuscular junction where a neuron meets your muscle. And that's a hot place to look at when it comes to the research and understanding the facilitation of movement for certain conditions. We'll talk about that. So the main flow of information is basically going to be from your cortex. We have regions in our brain that are kind of highlighted here, the premotor cortex where we plan movements. Really, if we start all the way from the prefrontal cortex, once you're a fully developed adult, you can think about or have an idea of some movement. That movement can get planned in the premotor area, which is Brodmann area 6, if you care. And then we can consciously stimulate that movement in the primary motor cortex, which is this highlighted green, this specific light shade of green here, which is also known as the precentral gyrus. This gyrus falls on the frontal lobe, by the way. So it's just before the central sulcus, in case you wanted a little bit of neuroanatomy there. And that motor cortex will send signals throughout other parts of your brain and then down through your spinal cord and then out towards muscles. Okay. I also want to kind of discuss that it's not just a one-way interaction. Sometimes when you go through movements, you get stimulus back. And that stimulus is a sensation. So you have sensory neurons that send signals back up to your brain to let you know if that movement was okay, or if it created tension in the tendons, or if it was painful, for example. And so this is a conscious back and forth between, or not so conscious, but a constant back and forth between the movements you do and the stimulus and experience that they create. Okay. So let's talk a little bit about muscles. When we're talking about voluntary movements, oftentimes we're mostly focused on skeletal muscle, but I did want to highlight that there are other kinds of muscles in our body that are all engaged by our nervous system. Some of them, like the smooth muscle, are engaged by our enteric nervous system, our GI tract, which is regulated by our vagus nerve. And some people like to say it has a mind of its own. So it kind of, there's a lot of really interesting research along those lines, and we'll discuss that when we get to reflexes. But your skeletal muscles, hopefully some of you by now have taken some anatomy. There are muscles here like the pectoralis major, the rectus abdominis, the quadriceps femoris. All of these muscles allow us to do a lot of movements. For example, the pec major allows us to bring our arms forward, which is horizontal adduction. And I like to think of it as like the hugging muscle. So whenever you go to reach for a hug, you're using your pecs there. And so all of these are skeletal muscles because they move our skeleton. Smooth muscles often are in our GI tract. And then there's a third called cardiac muscle, which is pretty unique. And that is just like it sounds in our heart, cardiac. When we're talking about voluntary, though, we're mostly talking about skeletal. And then there's also some other underlying reflexes that allow other reactions to go. So for example, when you consume food, your GI tract is working. That movement of what we call peristalsis allows the food to go through our entire GI tract. And maybe we can save that specific for a different lecture. Okay, let's take a look at just the skeletal muscle for now. So if you find your favorite muscle, a lot of people like the biceps brachii on the arm. That's usually a really easy example to go by. That biceps brachii connects from your shoulder, kind of your scapula, over your arm and onto your forearm. And that allows you to bend your forearm this way. So to kind of give you an idea is that when your brain stimulates your muscle, that muscle will contract and they will shorten. And when the muscle shortens, it's pulling on the bone, allowing you to cause a change in the angle of a joint or a change at the joint. Okay. So skeletal muscles are defined as muscles that attach to the skeleton and cause movements or actions on that skeleton, usually at a joint. And a single muscle, as you can see it externally like this on the screen, is actually made up of a bunch of fascicles. And then individual fascicles are made up of individual muscle fibers. And we can actually go deeper into myofibrils. And the point that I want to get across to you guys is imagine you had one rubber band. just a single rubber band, and you pulled it as far as you could, and then boom, it snaps. So there's a tension limit to one single rubber band. But if you put a bunch of rubber bands together and then wrap them in some sheath, and then put those bounded rubber bands against other copies of those, now you have this really, really strong fiber that can essentially multiply, or it's an additive kind of force to the strength of those rubber bands. And so when you think about muscle, think about individual fibers, kind of like individual strings of spaghetti. Instead of just one spaghetti, which can snap really easily, you have bundles and bundles and bundles of individual spaghettis wrapped together and bound together. And that's what kind of forms the muscle. And so that allows for a lot more strength and allow people to do crazy things. For example, run a marathon in 2 hours, which is a recent record that was broken, or to deadlift over 1,000 pounds, if you can believe it. So your muscles can be trained to be very strong, and you can actually train them to grow if that's something that you want. And that process is called hypertrophy. And so we can talk a little bit more about that some other time if you guys have questions. Another aspect about skeletal muscles that I really want to emphasize is because they attach to bone, those muscles have to be attached to this connective tissue called a tendon. So a tendon will connect muscle to bone. A ligament will connect bone to bone. Okay, so tendons are actually made up of a lot of connective tissue that you can actually see on the outside of these muscle fibers. This endomysium, which is wrapping around this fascicle, as well as the perimysium, as well as the epimysium, is this connective tissue that lies in multiple layers throughout this muscle belly and eventually forms this tendon. And it's very strong. In fact, it's more likely that you'll tear a muscle before you get a complete tendon tear. And as we'll see later in this presentation, if we have time, you'll see that there's actually sensory fibers called the Golgi tendon organ in the tendon themselves in order to sense that tension. And so if the tension is too large on the tendon, your reflexes will tell you to let go of the weight. It's too much, or to stop the stimulus from occurring. So if you imagine someone bench pressing or deadlifting or doing some type of movement, running, if the tension is too great and it stimulates the Golgi tendon organ going, this is too much, that'll quickly tell that organism to stop doing that thing that is causing the pain. Okay. A third point that I want to bring to your attention is that your skeleton has specific areas of attachment. And a lot of these parts of your skeleton, which I haven't highlighted in this presentation, but might be useful, are protrusions of bone that are kind of like porous looking. They look like they have like little indents, and they're very rough. These rough growths on the bone. The names of these things are called tubercles, tuberosities, and trochanters. There's actually those 3 different kinds. And tubercles are like the little small ones. It's like a little cute tubercle. Tuberosity is a big kind of large growth of bone where a muscle can attach to. Usually bigger muscles attached to these bigger growths. And then there's a trochanter. And a trochanter is like this big growth of bone that has a large surface area for that tendon to really grab onto. And so I can give you an example. I'll name one called the ischial tuberosity, which is your butt bone. If you kind of touch around your butt area, there's like this stiff bone where your hamstrings attach to. And those are very powerful muscles that require a really strong anchoring point to pull. They need that because if your muscle is really strong, but the connection between your muscle and the bone isn't, then you're just going to rip off the muscle from the bone, right? So the connection has to be very, very strong. Okay. So far, does anybody have any questions? Because I know I kind of rambled on a little bit there. Or are we doing good so far? If not, I'll assume yes, and I'll just keep going. Chilling like a villain. Everything clear? Great. Okay. So let's talk about muscle fibers. So every muscle fiber is controlled by a single motor neuron, but each alpha motor neuron can control multiple, potentially hundreds, if not thousands of muscle fibers. So it's not a perfect 1-to-1 ratio. It's a motor neuron can essentially recruit more muscle fibers. From a personal training standpoint, if I was not very regular at the gym, for example, let's say I couldn't do more than like a push-up or two, and I started going to the gym and I started doing push-ups, a bunch of push-ups once a week, twice a week for like 2 or 3 months. The amount of muscle that you can realistically grow in that time is not very significant, but the stimulus that you're giving to your body will actually train your body to recruit more muscle fibers, essentially allowing you to produce more force. And so you can learn to do more push-ups or more pull-ups relatively quickly, because you're better able to recruit those individual muscle fibers when you are trained to do so, rather than when you have no stimulus and no background training at all. And so in the gym, we call these newbie gains, because you're a newcomer and you're getting very strong very quickly. And then at some point that kind of levels off, and then a more realistic linear growth of strength is seen. But initially that recruitment is very important. So you can actually train to recruit more muscle fibers. And a practical example of this is if you look at people who lift very heavy weights for fun or for a living, power lifters as they're called. You don't necessarily have to be super big and bulky and have large hypertrophied muscles in order to lift a lot of weight. In fact, a lot of really strong power lifters are relatively skinny. Not that they're skinny in comparison to the general public per se, but they are skinnier than, say, a bodybuilder who trains for muscle hypertrophy. And the reason they're able to pick up potentially more weight as compared to a bodybuilder who is heavier than them, has more muscle than them, is because of the recruitment of muscle fibers and the training of specific movements, which makes the movements more efficient and therefore less strenuous. So there's a lot of really cool interplay here if you want to get into the field of physical therapy, physical rehabilitation, orthopedics, sports medicine, that kind of stuff. There's a lot of fundamental concepts here that you'll see if you go down that route. Okay. So, and when we're talking about the process of skeletal muscle activation, I kind of went over this. You have to think about this. The signal is sent through your central nervous system out through your peripheral nervous system. So that means that a upper motor neuron is sending a signal to an interneuron usually. An interneuron, or potentially just another neuron, is a lower motor neuron is going to send that signal from your central nervous system to the muscle. And then once that nerve eventually reaches the muscle, it's going to release acetylcholine, which is the neurotransmitter of the neuromuscular junction. And when that acetylcholine hits the muscle, it's going to cause a bit of a chain reaction that releases calcium. And there's a lot of details here that we'll potentially have time to go over throughout the year, but that will lead to muscle contraction. And we'll talk about how the muscle contracts at a different time. All right. So alpha motor neuron. So the alpha motor neuron, its function is to innervate the skeletal muscles that cause the contractions that allow you to move. This is something that I would note. I would make a note card out of this and memorize it. Skeletal muscles are only innervated by one alpha motor neuron, and they release the neurotransmitter acetylcholine at the neuromuscular junction. So just kind of reiterating what I already said. Now, do not get overwhelmed by this picture. I will make a dedicated video discussing the tracts of the human spinal cord. One thing I did want to mention, though, is to get you guys kind of seeing the structure and the organization of the human spinal cord. You'll see that the blue are sensory pathways. And one thing I want to highlight is that the sensory pathways are ascending, which means that they're going from your body up towards your brain. They're going up through your spinal cord into your brain. So they come from the outside. They stimulate your nerves, your sensory nerves in one way or another. That signal potentially, let's say the signal was at the tip of my finger, comes all the way to my spinal cord. And then that tract ascends up to my brain. So that's an ascending tract, and that's a sensory tract. Motor pathways are descending. They start in your brain, in your central nervous system, descend to your spinal cord, also part of your central nervous system, and then exit that. So they descend down and exit towards your body. You might have also heard the terms afferent and efferent used in this context. So motor pathways are efferent. They are exiting the central nervous system towards your body, whereas sensory pathways ascending are afferent. They're arriving. I like to think A for arriving and E for exiting. that might help you guys out. And some of these tracks are going to be very important to understand potentially in your future careers, future research, or maybe just for fun to understand. For example, the corticospinal tracks are the major tracks that allow for that voluntary movement of your skeletal muscle. You also have the sensation of touch, pain, temperature, vibration in the dorsal column pathway. And so we'll talk a little bit more in depth here, but I just wanted to give you an introduction into the spinal cord. Now, for the students that are here today, I do have a question. I want to engage with you guys. What kind of view of the spinal cord is this? How is the spinal cord laid out for us? Is this a anterior view, a posterior view? Has the spinal cord been cut in any way? If so, how? And if you don't know how to answer that question, you can simply say, I don't know, and I'll happily explain it to you. But I want to know what your thoughts are. And I want to give anybody who listens to this recording a chance to think this through. So we have a statement here. Is this anterior? Good guess. Good guess. Any other thoughts? So we are actually at the anterior. So this is known as the anterior median fissure. You're not going to have to know that at this point, but this, hopefully you can see my cursor. The cut of this is actually a transection. So imagine a spinal cord is this tube, maybe like this pen. If this was the spinal cord, I would cut it like this, like with scissors this way, and we're looking at it from a superior anterior view. So we're looking at it from the front and slightly above, and the spinal cord has been cut, transection, horizontal section. Just to give you an idea, this spinal cord should be a tube, and we're only seeing a really thin slice of that. Okay, so good job. I'm glad that you took the time to engage. Okay, so we're in terms of the motor unit, let's talk a little bit about the pathology that can happen. One of the things that you really need to know is something called ALS. And you may have heard of this from the ALS Ice Bucket Challenge. Amyotrophic lateral sclerosis is a disease that causes the death of motor neurons. Now, this can be upper motor neurons, which means in your central nervous system in your brain, or it can be lower motor neurons, or both. Oftentimes it starts lower motor neuron, it starts at the distal end, so like your fingers, your toes, and then eventually it can lead to paralysis overall. But amyotrophic, amyo means like muscle loss. So when your nerves, I think soon to be Dr. Bartfield mentioned that the use it or lose it mantra is important in sensation. It's also true in motor units. It's also true in movement. If your neurons, motor neurons, are not stimulating their end targets, for example, the muscles have no reason or stimulus to be present, and so they can atrophy. Atrophy, spelled atrophy, so like a trophy, but one word, atrophy, is the degeneration or decreasing of muscle. The full definition is a decrease in the cross-sectional area of the muscle. And that is the opposite of what bodybuilders do. Bodybuilders aim for hypertrophy, or hypertrophy as it's pronounced, which is an increased cross-section of the muscle. So if you don't use it, you lose it. And in this case, when the motor neurons die, the patient who is afflicted with this disease will also see significant muscle atrophy, which means that they will be less capable of performing movements. And that would potentially lead to involuntary movements or a lack of movement or paralysis overall. Okay, so I hope that is clear. And if you have any questions on those words and the definitions of them, and if you want me to repeat them, feel free to let me know. But that was atrophy. Okay. Another name for ALS you might want to know is Lou Gehrig's disease. But that's important. So amyotrophic, loss of muscle, lateral sclerosis. This is a sclerosis. Anytime you hear the word sclerosis, sclera or scleration or scarring, this is a degeneration of that tissue. So that's what that kind of translates to. So anyway, hopefully that makes a lot of sense. It is not the only disease of the motor nerves, but it is one common one that you're going to need to know for the brain bee. Okay. Now let's talk a little bit about joints. So joints are these incredible pieces of our anatomy that allow the rotation or the flexion and extension of our body. And really it's the joints that create the possibility of movement. Muscles actually perform the movement, but without a joint, there's really nothing to move. And you can see this, for example, in certain species of birds. They have fused thoracic skeletons, fused thoracic spine, and they don't actually have individual vertebra. And so there's no movement. They're very rigid. Think of like a turtle. A turtle can only like wiggle its hands and like its head, but its whole thorax is like this shell. And so it's not able to move. Now, that is beneficial for that species because it's a protective thing. I don't think you can't really herniate a disc if you don't have any. So the potential room for herniated disc nerve pain for turtles is probably very, very low, but they are not able to move in the same capacity that we as humans can. And so joints, for example, our spinal column are made up of many joints of vertebrae interconnected to each other with a spongy disc in between most of them to allow for movement. That allows us to use our muscles to kind of manipulate those joints in order to create the movements that we want. Okay, now I'm going to toss a couple of words at you guys that are very important. So when we are talking about the biceps brachii, there's actually another muscle in the back. You might have heard of it. It's called the triceps brachii, or just triceps if you prefer. Those muscles move along the elbow joint mostly. So when you look at the elbow joint, you can flex it or you can extend it. So in other words, you can push someone away with your elbow. Say you're pushing something away. Maybe you're shooting a basketball, like I like to do every now and then. Or you might do a bicep curl. So you might bring some weight up to you. If you don't like movements in terms of the gym, think about drinking a glass of water or picking up a burger to eat, which is probably a better, more relatable example because I love burgers and I know most of us do. So, whenever you're flexing your elbow, that is the biceps brachii, as well as some other muscles that we can mention later on doing that movement. Though the flexion at a joint is defined, so flexion at a joint is defined as the decrease in the angle of that joint. So if you look at my elbow, right now I'm 180 degrees. It's completely flat. But if I wanted to perform flexion at the elbow, that joint that's 180 degrees here can go from that to 90 degrees, 45 degrees, 30 degrees, or decreasing over the course. That is flexion. If the opposite was to occur, my triceps in this case are going to pull and increase the size of that joint. So I go from 30, 45, 90, 120, 180. And so that increasing angle of the joint is defined by extension. So flexion is a decrease of the angle at a joint. Extension is an increase of the angle at a joint. And there's other movements that we can talk about. So for example, adduction is actually going closer to the midline, your midline of your body. So if I bring my hands way out here and I clap in the center, what I just did is I adducted my arms together. I adducted my shoulder joints. Adduction. Abduction is the opposite. I'm being abducted by aliens, for example. I'm going away from the midline. I'm going away. Abduction. And that shoulder joint allows us to perform those things. And there's other movements like medial rotation, lateral rotation. If you have any questions on that, we can definitely discuss that. But for now, flexion, extension, adduction, abduction, all of those things are important to understand. I'll give you another example of flexion and extension. When you curl up into a ball, maybe you're, a movement like this is when you're working out your abs, you're flexing your spine. And when you wake up in the morning, or maybe you pick something up and you kind of stand upright, that is known as extension. So extension of the spine. So those are just a couple of examples. And they're usually spanned by what we call activator muscles. These are muscles that do the movements. And so this is essentially what I was trying to explain. Another example of extension and flexion, your quadriceps, they extend your knee, and your hamstrings flex the knee. Um, uh, and the hamstrings actually have other functions as well. And there's some nuance that we're not getting into here for time-wise, but you guys should definitely understand that muscles can manipulate joints. And oftentimes we have antagonistic muscles that work opposite of each other. And that opposite function is very important in order for us to be stable in a position. Okay. Anybody have any questions on muscles, agonists, antagonists, on joints, or on anything else that I've mentioned so far? Give a little bit of a reflection break here. And it's okay if you don't. You can say, I don't know, or you can say no questions. I'll give you another example while I look. No questions so far? Okay, great. One of the examples that we wrote on here is throwing a dart. I prefer the shooting of a basketball. I don't play darts. But when you go to throw a dart or shoot a basketball, not only are you, say, extending your elbow and potentially flexing your wrist, say for a basketball shot, flexing your wrist, extending your elbow, you put those two together, you get a nice fluid motion. If you only had that extensor of the elbow and that flexor of the wrist, there wouldn't be any playback to stabilize that movement, and you would just be flinging your joint towards a direction. And that can cause degeneration of that joint, potentially atherosclerosis later on. And so what the antagonistic muscles allow you to do is to really control that movement and finish it where you want. The antagonistic muscle is going to work against the motion in order to stop, to prevent joint damage. Okay, so the antagonistic and agonist-antagonist relationship is really important for stability, for the health of the joints, and for you to be able to do the movements that we often take for granted every day. Okay. So I already kind of explained this, so I'm just going to move on. So some muscles act on soft tissue rather than joints. The head and neck is the great example of this. So we do a whole ton of facial expressions. We smile, we laugh, we frown, we cry, we have movements in our eyebrows. Some of us can control them better than others. And these muscles don't actually act on any joints. They act on the tissue that makes up your scalp and your head, and allow for facial expressions. So this is also a relevant example. And I'll give you another question. Does anybody know the cranial nerve that is related to movements of the muscles of the face? So when it comes to facial expression muscles, does anybody know the cranial nerve that innervates those muscles? I'll give you guys a second. I'll give you a hint. It's pretty obvious once you realize it, but you have to know the cranial nerves for it to be obvious. If you don't know the cranial nerves, then I don't blame you. Any guesses? So the cranial nerve in question is cranial nerve number 7. Yeah, there you go. You got it. It's the 7th, and you know that because it's the facial nerve. The facial nerve innervates the movement muscles of the face, the facial expression muscles. Oddly enough, it doesn't innervate the sensation of the face. That's done by cranial nerve 5, but the movement of the muscles of the soft tissue is done by 7. Okay. All right, let's talk about involuntary movements. So an involuntary movement might sound like a bad thing. It's like, it's not your choice, it's not your decision, and now you might think of like flailing, and there are some conditions like Huntington's chorea where that flailing can actually be a pathology. It can be a medical thing. However, involuntary movements also refer to reflexes, and reflexes can save your life. And they can actually save your tissue. They can literally save your life. And they also, more recently in the literature within the last couple of years, we've noticed that the reflexes can actually manipulate the immunology of your body. You might think of the gut-brain axis. You might think of inflammation throughout the body. We know that the vagus nerve, for example, which is cranial nerve 10, is responsible for heart rate. And so that is a reflex. That is a stimulus where if your lungs are at capacity and they're full, when you take a deep breath, there's not enough space in your chest for a full contraction, and so your heart speeds up to compensate. And then when there's more space, it'll slow down. When you breathe out, your heart will slow down. That's controlled by the vagus nerve, and that's a reflex. And so there's a lot of reflexes that happen involuntarily that we take for granted, that we study to better understand medical conditions, okay? So just to name a few, I already mentioned Huntington's disease. Chorea is when you have this rapid, uncontrolled, large movement, but also Parkinson's and other ataxias can lead to tremors. So Huntington's disease in this case is also understood to be a hyperkinetic disorder that affects certain parts of the basal ganglia, specifically the striatum. So when we talk about the globus pallidus and the putamen, for example, that's usually related to Huntington's disease. And that'll be exemplified in a different lecture, but I did want to say that here so that future you'd be like, oh yeah, Sebastian mentioned it. Parkinson's disease is known as a hypo as in lower, hypokinetic disorder that affects the substantia nigra. The substantia nigra is translated to black substance, which is in your brainstem. There's actually two areas, at least that I know of, the pars compacta and pars reticulata. The pars compacta of the substantia nigra is the dopaminergic part of that. And if that is essentially injured and the cells there are dying, you can't produce the dopamine you need for those motor pathways, and therefore you can't control your muscle movements. Okay, so there's involuntary movements all throughout here. And now one involuntary movement that can save your life is one I experienced a lot of times as a kid. Sometimes I still experience it. In fact, I have a burn somewhere from when I was cooking something in the oven. So if you put your hand on something very, very hot, say a pot of boiling water, or the stove is very hot, or a grill, and you're a naive kid that has no understanding of the world, and your parents might tell you, don't touch that, it's really hot, you might be curious and say, well, what does that even mean? And so you put your hand on it, and what do? Immediately, almost immediately, you remove your hand. Not fast enough to prevent some damage from occurring, but fast enough to prevent significant damage from occurring. And that's the point. The reflex allows you to essentially not have to think about making a decision in order to prevent further damage to yourself. And this is done in this monosynaptic reflex example. That word I just used is monosynaptic, mono as in one, synaptic as in synapse. So it's just one synapse. It's a sensory neuron to a motor neuron. Sensory neuron screams like, oh my God. And then the motor neuron's like, whoop, we're going to move that. So that's the idea. So rapid response, highly myelinated motor nerve fibers allow this to happen so that the signal gets sent to you as well as out in the fastest way possible. And it prevents significant injury. So like if you didn't have this reflex, and I'm sure there's some medical condition that I'm not aware of where this might have happened, where a patient doesn't feel pain, for example. If you leave it there and you start to smell burning skin and you're like, oh, that's not good, I have to take it out. If you wait that long, now you have a ton of tissue damage and you might lose sensation or motor capabilities in your hands. You might start to bleed and that could lead to infection. And there's a whole lot of list of things that could happen that's bad if that is allowed to happen. But if you remove it fast enough with this reflex has evolved for millions and millions of years throughout mammals that you can prevent that damage from happening. Okay. Now, how can we use this in the clinic and how can we test you guys on this in the BrainBee? Well, there exists jerk reflexes. One of the most famous ones and most common ones used in the clinic is the knee jerk reflex. The knee jerk happens where the quadriceps, actually the tendon goes to your tibia. It attaches to something called the tibial tuberosity, to connect that to the last time. But there's a piece of tendon that sticks out just like basically below your kneecap, also called your patella. And between your kneecap and where that muscle attaches, there's tendon. And the physician or the medical provider can tap on that tendon using a little hammer, a rubber mallet, and can see that your leg will move. So your tendons, like I said before, have these Golgi tendon organs which sense stretch. And if they stretch too much, they will tell your body to contract the muscles so that the tendon doesn't get injured, because your tendon tendons are way more difficult to heal, but they're very, very strong. And so if you can contract your muscle and move the tendon and shorten it, you'll prevent more injury. Whereas if you have a lengthened tendon and you hit it, you could tear it. But if you bunch it up all together, it's much more resilient. And so our bodies have evolved this method of testing that out. You get a knee jerk because your quads contract, and that shortens the length of the tendon. That's called the knee jerk response or the knee jerk reflex. Now, if someone has an injury, for example, to the nerve that goes to their quadriceps or the nerve that connects their quadriceps and their tendon, and the provider hits the knee and there's almost no, they won't have a reflex. So if a patient comes in, you do this knee jerk reflex and there's barely any reflex, that could be an indication that they have some nerve damage where that nerve isn't appropriately responding to the stimulus. The opposite can be true. You can tap on that knee and then the knee can jerk erratically with very little control, and you can even get kicked if you're the person doing this. And that's a hyperkinetic response. That could be an injury to their brain. That can be an upper motor neuron lesion. And that can be indicative of many different things. It can be indicative of a tumor. It can be indicative of past stroke. It can be any cerebral injury that is related to that specific nerve tract can be related to that. So we use our knowledge of this neuroscience to be able to appropriately treat people in the hospital or in the clinic. And so that's why the fundamentals are very important to understand. Any questions on that while I zoom in on this? Preguntas? All clear? Awesome. Okay. Now, we actually don't just have a knee jerk reflex. There's also a biceps reflex. Any muscle that has a tendon that extend, there's a triceps reflex. You can test other muscles, but the most common one used in the clinic because of ease of access and just feasibility, practicality is the knee jerk reflex, okay? I do want you guys to understand that the receptors see that stimulus, they feel it, they have a change in their environment. That signal is sent afferent. Again, it's arriving to the central nervous system. It gets integrated, so that response basically gets sent back out almost immediately to a motor neuron, and the motor neuron tells your muscles to move, which gets you out of the way of that stimulus. Okay, so hopefully that is clear. So this is another name for this is the withdrawal reflex. You're withdrawing for that stimulus. And this often relates to nociceptors. Nociceptors are, this type of receptor that is specific for pain. So another interesting fact about this is that we have nociceptors in our mouth, and the spicy foods actually are not a taste receptor. It's a pain receptor. Which is why not a lot of people like it. But nociceptors are present throughout our body, and they sense pain, and so that is what this is related to. But anyway, we already went through this, so I'm not going to go through it again. Now, the muscle spindles, this is what I was talking about before. They sense stretch in the muscles, and we also have this in our tendons. And so if there's too much stretch, for example, in our muscles, or too much stretch in a short amount of time, that can lead to a reaction that causes you to say, let go. So I've experienced this in the gym, for example, while I'm kind of fatigued doing whatever it is that I'm doing. And I get to the point where I'm so fatigued that I feel like, oh, I just stretched something really bad, and you let go of whatever you're doing. It's like a reflex of like, oh, stop. And it works through the same kind of pathway. Your voluntary movements or involuntary movements, they sense the stretch as well as the speed of the stretch. And that'll stimulate a sensory neuron, which sends a signal to your central nervous system, and then can send a signal back out to the motor reflex to essentially manage that reaction. Okay. I do want you guys to take some time to look at this diagram, look where the muscle spindles are and in relation to the muscle bellies themselves. And there are a lot of really good YouTube videos online for this. Okay. So the other thing that we need to understand, and kind of moving this to another level, is you can have a reflex, but the hyperkinetic aspect of it, remember when I talked about agonists and antagonists, the agonist will cause the motion, but the antagonist is also partly responsible for controlling that movement. So when you're looking at the knee-jerk reflex, you're not just looking about their sensory systems, you're also kind of indirectly looking at their hamstrings and the nerves that go there. So there's actually two sets of nerves. It's not just your femoral nerve, it's also your tibial nerve, which comes from your sciatic nerve. So there's all these relationships that are at play. And I also included a couple of fun facts here, which is that you can actually train yourself to override this. You have the conscious thought that allows you to kind of like get rid of this. And here's kind of a better diagram to show this. So what I want to show here is give you a visual of what I was explaining before. This bone here is called the tibia. The other bone here that we can't really see is called the fibula, but the tibia is this kind of major bone here in the lower leg, and that connects to the patella here. The patella actually isn't fixed on it. It's actually connected inside of the tendon to the quadriceps. It's known as a sesamoid bone, so it actually grows within the tendon. It doesn't actually connect strongly to any other bone. It lives inside of the tendon. And then here's the femur, which is home to the quadriceps and the hamstrings, kind of, that's where the muscle bellies of these lie, is along the femur. And when you hit this tendon, which is present between the patella and the tibial tuberosity, this part is known as the patellar tendon. That is what is being stimulated, causing the stretch receptor to be notified that, oh, this is being stretched, we need to contract. And that's why the leg moves up. As the leg moves up, the distance that patellar tendon takes between the patella and the tibial tuberosity is lessened, and that is a much more rigid structure. Okay. All right. Let's see. All right, so feedback systems. So as movement occurs, muscles provide feedback to the brain, and that information in terms of your body's orientation, in terms of the muscles you're using, in terms of the task that you have in mind, will all be kind of put together into one piece, and this can actually be trained. So think about shooting a basketball. If you're shooting a basketball, you're not just moving one muscle, you're moving many, many muscles, you're moving many, many bones, and you're working through this motor pattern. And your muscles will move at different speeds, and you can choose to move them at different speeds, either generate more power or less power. And so your body has this feedback system to basically integrate that information to improve on the movements that you've been performing. Okay. So step 2 is those muscle spindles inside. Once you're moving, they will sense the stretch, and that part also gets integrated into the movement pattern so that you are not hurting yourself throughout the movement, and that the movement is performing the task that you wish to perform. Step 3 is the gamma motor neurons. Essentially, you adjust the sensitivity of the system via a different pathway, and that movement gets sent back. So as you're doing the movement, you're getting constant feedback from the sensory stimulation, and that can adjust movements moving forward. And that's kind of the basic idea here. Does anybody have any questions on anything that I mentioned? Because I realize that we're getting really close to time, and I don't want to overload you guys with information because there's a lot of text. All good so far? All good? Is Ina the only one in here? Let me see. Any other students in here? Oh, we have some other people here as well. Okay. So to kind of continue a little bit here, I have already mentioned the Golgi tendon organ, but step 4 is that we need to ensure that our tendons stay stable and that they're not overstretched. Some of the most common injuries in sports, for example, ballet, football, basketball, there's probably a bunch of other ones I'm missing, is the Achilles tendon. That Achilles tendon is very, because it's responsible for so much weight, so much movement, and especially at these higher level of sports, the strain on them is very large. That can cause the tendon to tear, especially if not enough time is allowed for recovery. That is a much longer healing time than, say, just tearing a muscle. So on the scale of this, you can have small muscle strains, you can have muscle tears, and in muscle tears, there's a spectrum. You can have a complete muscle tear, and then after that, you can potentially have, not necessarily after experiencing that, but those are a set of possibilities. Another possibility that is less common is a tendon rupture or a tendon tear. That takes a significantly longer time to heal and usually more surgical intervention than muscle tears and strains. Strains usually don't require any surgical intervention whatsoever, just some physical therapy. And the reason we're able to sense this most often than not before they happen is thanks to the nervous system that's in play between our muscles as well as in the tendons. All right, so complex movements. So this is where we kind of get into the spinal cord circuits. You're never really just moving one muscle. Just speaking to you requires me to move many muscles of my face and my mouth in order to accomplish the task of simply communicating to you. And that means that different muscle fibers from the same cranial nerve have to be stimulated different times and in different ways. And that is constantly being sent back into feedback into your brain and spinal cord, which is allowing you to better actuate that. So I can give you more examples of like motor movement patterns, but another thing is, for example, singing. So professional singers have a lot of training under their belts that allow them to move their vocal cords. You have muscles that control your throat, your vocal folds, as well as other aspects that allow you to make noises in different particular ways. And these feedback loops, which are very complicated, require different muscles, allow you to get better at those movements. So different brain regions will also get stimulated depending on what kind of movements you're doing. And you can coordinate those. And so you not only do you have your motor cortex, but whenever you're doing volunteer movements, you also have to remember that there's basal ganglia at play. There's also your cerebellum at play. And so imagine it's not just one brain region to one muscle, it's multiple muscles, multiple brain regions, and all these connections are working together in tandem to process sensory information as well as send out new motor information. So just to keep that in mind, and there's a couple of different articles here that we highlighted, so that when you go home and read this on your own, you can kind of do some further investigations. Okay. Probably I said that I wasn't going to get into this yet, so I'm going to stop right here. Do you guys have any questions on today's lectures, on today's topics? Anything you guys want to be clarified? Hopefully that was a decent introduction into chapters 2 and 3. Great. I'm glad you guys like the visual. Some of these were present from older BrainBee PowerPoints, and then some of these are I mean, BioRender is really taking over. I mean, really, really good images, really, really good figures.
I'm currently on BioRender.
It's a cheat code. It really is. All right.
Sebastian can help me with some of my figures that I've been struggling to make on BioRender.
It helps that I have a Photoshop certification. So whatever you can't make in BioRender, let me know, and I can try to put it out in like in some other way. I can draw stuff and then like send it out. You got to give me a shout out though in your presentation or whatever it is. Be like #thanks Seabass or something.
Put it as it's for a manuscript. I'll put it as the figure title. #thing. I'm sure my PI would love that.
#thanks Seb. Love it. All right. Well, if you guys don't have any questions, I think I missed the part about where we were kind of formulating that resource for them to ask questions and then us to get back on those questions later. But hopefully we'll get that set up soon.
I think the way we'll do it going forward is that we'll have a slide. Hopefully, we'll be able to have a larger group of students. We were so appreciative of the students that could come today, but I know one school was not able to come. But at the beginning of presentations going forward, I'm going to try to get it set up that we can have a Slido, so students will receive a code that they can go and enter on the website, and then they can type in anonymous questions, or they're always welcome to use the chat if they don't want to ask in the chat. They can just send them in anonymously. But we will be sending the PowerPoints out ahead of time, so I think within the next week or so we can send, hopefully, I'm hoping we can send the teachers the PowerPoints for next month, and then students will have this month until then to go over it, and then we'll come hopefully with questions to our next meeting and have some good questions for us, have some good discussion. We are so excited that you guys are joining us and are doing the BrainBee, and we couldn't be more excited to be hosting a BrainBee, so thank you.
And next month we'll have the lecture given by Dr. Rosenberry, who's faculty here at Mayo, and he's also going to do a guest lecture presentation on his own research as well. So that'll be exciting. And I know we weren't able to cover chapter 4 today, so if you guys can maybe look at that on your own and then come with questions for the next session, we definitely can cover those questions. So please, if you can go over that and send it in to us, that would be great. And of course, I'll send out that PowerPoint if I haven't already. I can't remember now. I'll send out the PowerPoint for all the chapters.
So thank you all for coming to our inaugural Be-Wide Zoom session. I rate it a success.
Thank you guys. I'll send the recording.
I second that.